You know, when we sit back and reflect on our own lives, there is this really profound, almost irresistible temptation. We tend to view the human brain as this flawless
Oh,
archivist.
absolutely. We really do.
I mean, we like to imagine there is this meticulous little librarian inside our heads, working in this vast, perfectly climate-controlled, dustless room.
Just carefully filing away every single thing that happens.
Right, exactly. Every conversation you have ever had, every sunset you have watched, or heartbreak you have endured. We think of them being slipped into these neat, chronological manila folders.
It is an incredibly comforting illusion, though, isn't it?
It really is.
Because we just operate under this everyday assumption that the file is always sitting right there on the shelf, perfectly preserved, exactly as it was written on the day the event actually occurred.
Just waiting to be pulled down and read.
Yeah. But then you look at the actual architecture of the brain. You look at the rigorous cellular reality of how human experience interacts with biological tissue.
And that quiet librarian completely vanishes.
Completely. Suddenly, you aren't looking at a passive archive at all. You are looking at a loom that is constantly, relentlessly unraveling and reweaving its own threads.
It is the ultimate dynamic landscape.
It really is. Because what modern neuroscience reveals is that every single time you access the past, you are not simply reading a static document. You are actively altering it.
Which is wild to think about. You are rebuilding that memory from scratch in real time.
Yes, you are reconstructing it, which leads to this incredible realization that our memories are actually as much about the present moment and the future as they are about the past.
Which brings us perfectly to our mission for this deep dive. We're embarking on a really rigorous masterclass level exploration today.
We are diving deep into Part 8 of the foundational text principles of neuroscience.
Specifically, Chapter 52, which is learning and memory. Our objective here is to understand the absolute miracle of how experience literally becomes biology.
Exactly. We want to know how a fleeting, temporary moment in the outside world permanently alters the physical cellular structure of your nervous system.
It represents one of the most profound bridges in all of science, the physical bridge between neural activity and your inner mental life.
Because think about it. If your nervous system could only respond to the immediate present moment, your behavior would be entirely rigid.
You would just be a biological automaton.
Right. You'd be trapped in this endless loop of reflexes. So learning is the specific mechanism that allows experience to modify the brain.
And then memory is what allows those biological modifications to persist over time so they can actually influence how you behave tomorrow.
Exactly.
Okay, let's unpack this right at the foundation. Because we casually toss these words around every day, like learning and memory. But in a clinical neuroscience context, they mean very specific, distinct things.
They definitely do. They are not interchangeable.
So can you lay out the exact definitions for us?
Sure. So in a neurobiological context, learning is strictly defined as the experience-dependent change in behavior knowledge or behavioral potential.
So it is the acquisition phase.
Yes, it is the exact moment the brain takes in new information and modifies its circuits.
Okay, and memory.
Memory, on the other hand, refers to the entire suite of processes by which that acquired information is encoded, stored, and eventually retrieved.
So to distill it down, learning is the active process of change in memory, is the retention and later expression of that specific change.
You nailed it.
I want you listening to this to pause for a second. Truly imagine what happens if that retention completely disappears.
It is a terrifying thought experiment.
It really is. If the capacity for memory just vanishes overnight, what are the actual stakes for a human being?
The stakes are literally your entire existence as an independent, coherent entity.
Wow.
A life without memory is not just a life without nostalgia. It is not just missing the fond reminiscence of your childhood.
It's much deeper than that.
It is the total devastating loss of psychological continuity. You lose the ability to recognize the faces of the people you love most.
You lose the ability to navigate your own home right.
Right. Or to execute basic learned motor skills. You couldn't comprehend language or maintain any sort of continuous autobiographical identity.
You just become completely trapped in an ever-changing, terrifyingly disconnected present.
Exactly. You literally couldn't connect the sentence I am speaking right now to the sentence I spoke 10 seconds ago.
But you wouldn't know which foods are safe to eat or which environments pose a threat.
Or who you are in relation to the world. So memory is an absolute non-negotiable requirement for independent survival.
Now, historically, figuring out where this vital survival mechanism actually lived in the brain was a massive headache for the scientific community.
Oh, it was a tremendous puzzle.
Yeah.
Because by the 19th century, early neurologists like Paul Broca and Carl Wernicke had made huge strides.
Right. They had successfully identified specific localized brain regions.
Yes, regions that were absolutely critical for distinct functions like language production and motor movement.
But memory just completely defied localization.
It did. Whenever early researchers looked for a single dedicated quote-unquote memory center in the brain, they came up empty.
It just seemed to be everywhere.
Right. Memory storage appeared to be scattered, involving many different regions simultaneously.
Which is frustrating for researchers.
Extremely. It actually led some very prominent early scientists to throw their hands up and doubt whether memory was even a discrete biological function.
They thought it might just be part of general perception.
Exactly. But modern cognitive neuroscience has totally shifted that paradigm.
Because we know now that memory isn't just a mystical force floating everywhere, but we also know it isn't sitting in one tiny, neat little box in the cortex.
That is the critical breakthrough. Today we understand that memory is widely distributed, but it is distributed across highly specialized, anatomically distinct neural circuits.
Different kinds of memory rely on entirely different cellular systems.
Right. So to truly map out this incredibly complex network, we have to look at memory along two major dimensions.
Okay, what are they?
The time course of the storage, meaning how long it lasts, and the actual nature of the information being stored.
Okay, let's dive right into that first dimension. The time course. We are talking about the journey from transient, flickering biological buffers to durable, lifelong physical traces.
It is quite a journey.
The text breaks this down into short-term memory, working memory, and long-term memory. Let's start with short-term. Functionally, what is happening there?
So short-term memory is essentially a transient, highly fragile representation of information. And it is strictly relevant to your immediate goals.
Okay.
It allows information to remain actively available in your mind for just a few seconds after the original sensory stimulus has disappeared.
So classic real-world example would be someone telling you a verification code for your bank account.
Exactly. You hold those 6 digits in your mind just long enough to type them into your phone.
Or when you are listening to a complex sentence, you have to keep the first half of the sentence active in your short-term buffer just to comprehend the grammatical conclusion.
Right. It is a temporary holding space, and it degrades rapidly.
Now, people often use short-term memory and working memory interchangeably, but in neuroscience, they're definitely not the same thing.
No, they are fundamentally distinct.
I like to think of it like this. If short-term memory is just a passive holding tray where you set a glass down for a second, working memory is the chef's cutting board.
That is a brilliant way to conceptualize it.
Because it isn't just sitting there. You are actively chopping, mixing, and manipulating the ingredients to create a meal.
Working memory is an active, highly dynamic mental workspace. It is the system responsible for not just maintaining information, but manipulating it, updating it, and crucially protecting it from outside distraction in the service of whatever behavior you are currently executing.
So when you are reasoning through a complex math problem in your head, you are using working memory.
Yes. Or planning your driving route to avoid traffic, or comparing two different arguments.
Because it requires this active manipulation, it has to be tied to attention, right?
Absolutely. It is inextricably tied to attention and the prefrontal cortex's executive control systems. It is the memory system of the now.
And then we cross the critical threshold into long-term memory.
Yes.
William James, the pioneering psychologist, famously referred to this as secondary memory. He called it the knowledge of a former state of mind after that state has completely dropped from conscious awareness.
It is a beautiful definition.
But transitioning from that active cutting board to the deep freezer of long-term memory, that requires some serious biological heavy lifting, doesn't it?
Oh, it requires a profound physical transformation of the tissue.
Because short-term and working memory generally depend on just sustained patterns of electrical neural activity.
Right. They rely on very temporary, fleeting changes in the chemical efficacy of a synapse. It is basically an electrical hum.
But long-term memory is different.
Very different. Long-term memory requires durable biological modifications.
Yeah.
At the cellular level, we are talking about long-lasting synaptic plasticity.
So what does that actually mean physically?
It means the brain actually has to alter gene expression, synthesize new proteins, and physically grow new structural connections.
Like actually growing new branches.
Yes, dendritic spines. It grows new connections between neurons. The actual architecture of the brain physically changes its shape to hold that long-term trace.
This raises an important question regarding capacity, though, because if working memory is just an electrical hum and long-term memory is a physical restructuring, how much can these systems actually hold?
The contrast is staggering. Working memory is incredibly powerful in its processing ability, but its capacity is strictly and notoriously limited.
You can only hold a few items, right?
Yeah, maybe 4 to 7 chunks of information on that cutting board at one time before things start falling off the edge.
And long-term memory.
Long-term memory, conversely, appears to have an absolutely enormous, practically limitless capacity to store knowledge, skills, and decades of life events.
Wow, limitless. So we never just fill up the hard drive.
No, the main limitation of long-term memory isn't that you run out of physical storage space.
Yeah.
The limitation is access.
Ah, it is the problem of retrieval.
Exactly. The information might be physically stored in the tissue, but you might not be able to find the specific neural pathway to access it when you need it.
And this brings us to one of the most critical bottlenecks in all of human cognition, the transfer of information from that transient limited workspace into the durable long-term space.
This is a huge point.
I really want to emphasize this to you, the listener, especially if you are trying to learn a new skill or study for an exam. Simply being exposed to information does not equal encoding.
It definitely does not. Staring blankly at a textbook page or letting a lecture wash over you while you scroll your phone is just exposure.
Exposure is not learning.
That is perhaps the most fundamental neurobiological truth for any student to grasp.
Because we are bombarded by a massive continuous fire hose of sensory information every waking second.
Yes, but only a microscopic fraction of that data is ever stabilized into long-term memory.
Why is that?
Because selective transfer is biologically expensive and incredibly demanding. It requires focused attention, emotional relevance, motivation, and a deliberate effort.
You have to relate the new information to your prior knowledge.
Exactly. If you do not actively engage the prefrontal machinery of meaning and attention, the information simply decays from the short-term buffer in seconds.
And it is lost forever.
Forever. The brain is highly economical. It is not going to spend the metabolic energy to synthesize new proteins and grow new synapses unless it has been convinced that the information matters.
Matters for your future survival.
Right.
Okay, so we have mapped out how long memories last and the biological cost of transferring them. Now we must transition to the second dimension, understanding exactly what these memories contain.
Yes, the content dimension.
And to explore the nature of the information being stored, we have to talk about a historical medical breakthrough, a case study that fundamentally rewrote the textbooks.
We are talking about the critical division between explicit or declarative memory and implicit or non-declarative memory.
And this entire framework is anchored by arguably the most famous and important patient in the history of neuroscience, patient HM.
Henry Molaison.
Yes. Before we introduce HM and his specific tragedy, let's clearly define the two sides of this memory coin. What exactly is explicit memory?
Explicit memory is memory for knowledge that you can consciously access and verbally report. You can, quote-unquote, declare it.
Okay.
And is further broken down into two highly interactive subcategories, semantic memory and episodic memory.
Let's start with semantic.
Semantic memory is your memory for objective facts, concepts, categories, and meanings. It is knowing that Paris is the capital of France.
Or knowing that a neuron is a brain cell.
Right. It is completely detached from your personal experience of learning it.
And episodic memory.
Episodic memory, on the other hand, is the memory for personally experienced events anchored in a specific time and place.
So it is remembering when you visited Paris.
Exactly. The specific smell of the bakery you walked past on a Tuesday morning and the exact conversation you had over coffee.
And it is fascinating how those two systems feed into each other, because your episodic experiences gradually build up your semantic knowledge over time.
They do?
Like you might attend 50 different lectures on biology, right? 50 distinct, vivid episodes. Over the years, the memory of the specific lecture halls or the professor's voices fades away.
The episodic context dissolves.
Right, but the semantic knowledge, the hard facts about cell division, that remains perfectly intact.
Yes. And the relationship works in reverse, too.
How so?
Your existing semantic knowledge heavily dictates what you are actually capable of encoding when you enter a new episodic situation.
Oh, that makes sense.
Yeah, if you walk into a chess tournament and you have no semantic knowledge of the game, your episodic memory of the event will just be a blur of wooden pieces.
But if you are a grandmaster, Your semantic framework allows you to encode highly specific episodic memories of incredibly complex board positions.
Exactly. They are distinct systems, but deeply, deeply intertwined.
Now, on the other side of the dividing line, we have implicit memory.
Right. Implicit memory refers to the diverse forms of memory that guide your behavior without any conscious awareness or need for conscious recollection.
So it is memory expressed entirely through performance.
Performance, habit, motor skills, or physiological responses. When you ride a bicycle, you aren't consciously recalling the physics of balance.
Your motor system just knows.
Right. You are showing that your nervous system has changed, but you don't necessarily know that you know it.
Which brings us to the man who proved this division actually existed in the physical brain. Patient HM later revealed to be Henry Molaison.
A truly tragic but incredibly important figure.
In the 1950s, Henry suffered from debilitating, absolutely life-threatening epilepsy. And to save his life, neurosurgeons performed what was then an experimental procedure.
A bilateral resection of his medial temporal lobes.
They literally suctioned out the medial temporal structures on both sides of his brain, including the hippocampus.
Yes. The surgery successfully controlled his seizures, but the unintended consequence was profound anterograde amnesia.
He completely lost the ability to form any new long-term explicit memories.
It is a profound medical tragedy that honestly birthed modern memory science.
Because from the moment Henry woke up from that surgery in 1953 until the day he died in 2008, he was essentially frozen in time.
He was. He could not remember the names or faces of the researchers who worked with him every single day for decades.
He could not remember what he had for lunch an hour ago.
He couldn't learn new vocabulary words that entered the cultural lexicon after his surgery.
Wait, I want to push back on this, or at least play the skeptic for a second because it sounds paradoxical.
Okay, go for it.
If his memory was totally destroyed, how could he hold a normal conversation with the doctors? How did he even know how to speak or function in the room without just staring blankly at the wall?
And that skepticism is exactly what led to the breakthrough.
Eh.
Because you are right, his memory wasn't totally destroyed, it was selectively destroyed.
Selectively.
His working memory, his language skills, his personality, and his baseline intelligence were all completely spared.
Because they live in different brain regions.
Exactly. Because his working memory was preserved, he could easily hold a highly sophisticated, engaging conversation with you in the present moment.
He could retain the thread of the dialogue on his mental cutting board.
Yes. But the second you left the room, his attention shifted, and that transient working memory trace vanished.
So when you walked back in 5 minutes later, he would have absolutely no explicit recollection of ever meeting you in his entire life.
None. The bridge between the transient working buffer and durable explicit storage had been physically severed.
But the breakthrough goes even deeper than what he lost. It is really about what researchers discovered he could still learn.
This is the most amazing part.
They sat him down and had him perform a highly specific visuomotor task, tracing the outline of a star while looking only at the reflection of his hand in a mirror.
If you have never tried this, it is incredibly difficult and awkward.
Oh yeah, your brain wants to move your hand in the opposite direction. It fights you.
It is deeply counterintuitive. But over a period of several days, the researchers had HM practice this mirror tracing task.
And day by day, his performance improved dramatically.
He made fewer errors and completed it faster. He displayed a learning curve perfectly identical to a neurologically intact person.
His hand learned the skill. His motor system adapted beautifully.
But, and this is the astonishing part that changed neuroscience forever, every single day he sat down in front of the mirror. He genuinely, verbally insisted he had never seen the apparatus before in his life.
Wow.
He had zero conscious, explicit memory of ever practicing the task. Yet his implicit motor performance proved unequivocally that his nervous system was successfully storing the new information.
That one single case study demolished the idea that memory is a single unified faculty.
It proved beyond a shadow of doubt that the nervous system has parallel tracks. It can powerfully store experience without the conscious mind having any access to it whatsoever.
It was a monumental paradigm shift. It proved that the medial temporal lobe is absolutely essential for the formation of new explicit conscious memories.
But it is completely unnecessary for the acquisition of new implicit motor skills.
So Henry's surgery perfectly isolates the medial temporal lobe as the biological engine of explicit memory. Let's make that our next focal point.
Okay.
What exactly is happening inside the medial temporal lobe that allows a complex moment in time, a birthday party, a car accident, a first date, to be captured?
We need to look at the anatomy and the 4 distinct stages of episodic memory.
Let's start with the anatomy. The medial temporal lobe, or MTL, is highly specialized, right?
Very. It consists of the hippocampal formation and a funnel of adjacent cortical regions that feed information into it.
The entorhinal cortex, the parahinal cortex, and the parahippocampal cortex.
Yes. And geographically and functionally, these structures are positioned at the absolute apex of the brain's cortical hierarchy.
Meaning they don't receive raw, unprocessed sensory data?
Exactly. They receive highly processed, highly synthesized sensory and cognitive information from widespread association cortices all over the brain.
So to use an analogy, the hippocampus isn't a hard drive storing a single video file of your memory. It is more like a master film director.
I love this analogy.
You have the visual cortex acting as a camera crew shooting the visual scene. You have the auditory cortex recording the sound mix.
The amygdala acting as the composer, laying down the emotional soundtrack.
Right, and these are all happening in different cortical studios across the brain. The hippocampus acts as the director, stitching together those separate audio-visual and emotional elements into a single, unified, coherent scene.
That is wonderfully precise. The technical term for what your director is doing is relational binding.
Relational binding.
An episodic memory is not a single object. It is a complex, structured relation among many different, widely distributed elements.
The who, what, where, when, and how you felt.
Exactly. The hippocampus doesn't store the visual details of your grandmother's face itself. It stores the index, the relational map, that binds those distributed cortical elements into a coherent episode.
And this massive undertaking happens in 4 distinct sequential stages: encoding, storage, consolidation, and retrieval.
Let's break those down. First up is encoding.
And as we mentioned earlier, with the transfer bottleneck, encoding is not just being a passive sponge soaking up water.
No, it is a highly active metabolic process.
There is a massive measurable difference between deep encoding and shallow repetition.
Deep encoding requires you to actively focus on the meaning of the information rather than just its superficial sensory features.
So if you are trying to memorize a list of words and you just read a word over and over, analyzing its font or how many vowels it has, that is shallow encoding.
Yes, and the memory trace will be weak. But if you think about what the word means, how it relates to other concepts you already know, and why it matters to your life, that is deep encoding.
This deep strategic control of information relies heavily on the prefrontal cortex, right?
Yes. While the parietal cortex simultaneously directs your attention to the most salient features of the environment.
And what's wild is that neuroscientists can actually see this depth of processing happening in real time using functional imaging.
Oh yeah, the subsequent memory effect.
It's called the subsequent memory effect.
Yes, it is one of the most reliable findings in cognitive neuroscience. If we put someone in an fMRI scanner while they are learning a list of items, we can look at their brain activity during the exact moment they study each specific item.
And then later you take them out of the scanner and test their memory.
Exactly. When we look back at the scans, we see a clear pattern. The items they successfully remembered later were associated with significantly higher bursts of activity in the prefrontal cortex and the hippocampus during that initial learning phase.
Compared to the items they subsequently forgot.
Right. The depth of the biological engagement at the precise moment of encoding perfectly predicts the future success of the memory.
Once it is successfully encoded, it moves to the second stage, which is storage. And we've touched on this, but it bears repeating because it is so counterintuitive. Storage is heavily distributed.
Very distributed. The memory of a roaring campfire isn't sitting in a dedicated campfire neuron inside the hippocampus.
Not at all. The visual aspects of the flickering orange light are stored back in the visual association cortex at the back of your head.
The crackling sound of the wood is stored in the auditory cortex on the side of your head.
The smell of the smoke involves the olfactory system.
Right. The memory is distributed across the very same neocortical systems that originally processed the sensory input in the first place. Storage is a network-wide phenomenon held together by the hippocampal index.
Which brings us to the third stage and arguably the most complex to conceptualize: consolidation.
The stabilization of a highly labile, fragile new memory trace.
But the text is very clear that there are 2 entirely different types of consolidation happening: cellular and systems.
And they operate on entirely different timescales. Cellular consolidation happens rapidly over a period of minutes to hours.
This involves the local molecular changes we mentioned earlier, gene transcription, protein synthesis, and the physical strengthening of the specific synapses directly involved in the memory trace.
It is basically securing the local wiring. But systems consolidation takes much, much longer, days, months, or even years.
Wow, years.
Yes. This is a progressive, macro-level reorganization of the memory trace across the entire brain. Over time, through repeated replay of the memory, which heavily occurs while you are sleeping, by the way, the connections between the diverse neocortical regions become incredibly strong.
Oh, I see. They become so strongly linked to one another that they eventually no longer require the hippocampus to act as a director to bind them together.
Exactly.
I want to clarify that for the listener because it is a very common misconception. Systems consolidation is not just moving a memory from the hippocampus to the cortex.
Like dragging a file from a USB drive onto your desktop.
Right. It is not that.
It is definitely not a geographical relocation. The elements were always in the cortex. Systems consolidation is a progressive, structural reorganization of the network itself.
The scattered cortical nodes become structurally and independently linked to one another.
And once that network is solidified, the memory is fully consolidated. And it frees up the hippocampus to focus on binding entirely new relational episodes.
And the final stage of the episodic journey is retrieval. And if there's one single concept I want you to take away from this entire deep dive, it is this. Retrieval is reconstructive.
It is not a perfect high-fidelity video replay.
When you retrieve a memory, you are absolutely not pressing play on a finalized lockdown recording. You are actively reconstructing the event on the fly.
You are pulling from fragmented traces. And crucially, that reconstruction is heavily influenced by your current expectations, your current emotional state, and your immediate goals.
The brain uses the relational index to grab the visual, auditory, and semantic pieces, and it rapidly, sometimes messily, stitches them back together in real time.
But what is absolutely fascinating here, and frankly a little mind-blowing, is how this exact reconstructive machinery is used for something completely different.
Imagining the future.
It is one of the most profound insights in modern neuroscience. Think about it. The episodic memory system did not evolve simply so we could sit back in our armchairs and nostalgically reminisce about the good old days.
Evolution doesn't care about nostalgia.
It evolved to allow us to simulate possible futures to ensure survival. When you imagine a future scenario like picturing how a job interview might go next week or imagining navigating a foreign city you have never been to, you're actively pulling apart the fragments of your past experiences.
And recombining them into novel, simulated scenarios.
You're using the raw materials of the past to predict and prepare for the future.
Which perfectly explains why patients with severe episodic memory impairment, like those with extensive hippocampal damage, don't just lose their past.
Right. They stare into a blank void when you ask them to imagine what they're going to do tomorrow.
They cannot picture themselves in a future scenario. They cannot mentally time travel in either direction. The fundamental machinery of time travel is broken.
Exactly. Memory is fundamentally a tool for adaptive, future-oriented decision-making.
Okay, so the medial temporal lobe is doing this incredibly heavy, metabolically expensive lifting, organizing our explicit relational episodes so we can consciously navigate time.
But what about all the automated behaviors running quietly in the background?
Exactly. If we had to consciously remember how to walk or how to reflexively hit the brakes when a car stops in front of us, our working memory would be overloaded and we would crash.
There has to be a shadow system.
This brings us to section 4. Implicit memory, the body's hidden knowledge.
Implicit memory is a massive, highly diverse category. It encompasses several completely distinct neural mechanisms,
including priming skill learning, habit formation, and basic conditioning.
These are the systems that allow your body to act intelligently and adaptively without burdening your conscious working memory workspace.
Let's start with priming. Priming is essentially the automatic, unconscious influence of a past exposure on your present behavior.
But the source material draws a really sharp, fascinating line between conceptual priming and perceptual priming.
Yes, and they operate very differently.
Conceptual priming enhances your access to semantic knowledge. For example, if you have recently been having a deep conversation about oceanography, your brain temporarily lowers the activation threshold for related concepts like tides, whales, or salinity.
Interestingly, functional imaging shows that conceptual priming is actually linked to decreased activity in the left prefrontal cortex.
Way decreased. You would think it would be more active if it is primed.
You would think so, but the brain actually expends less metabolic energy to process the related concepts because the neural pathways have already been warmed up, so to speak.
Oh, that makes total sense. It reflects a highly efficient, streamlined processing state.
And perceptual priming is a completely different beast. It is tied to a specific sensory modality, right?
Yes. If you see the visual shape of a specific word, Your visual cortex is primed to recognize that exact physical shape faster next time, regardless of what the word actually means.
And there is an incredible double dissociation here with amnesiac patients that proves this.
This is a classic neuroanatomical proof of distributed systems. Amnesic patients with completely destroyed medial temporal lobes who cannot form conscious memories show perfectly intact perceptual priming.
Wait, really?
Yes. If you show them a list of words and later show them highly fragmented, degraded images of those words, they can identify them much faster than words they haven't seen.
They show the priming effect perfectly, even though they will swear to you they never saw the original list.
But if you test a patient who has an intact hippocampus, but specific localized damage to their visual sensory cortex, you see the exact reverse.
Oh, wow.
They have perfect explicit memory of the testing session. They can tell you every word on the list, but they show zero visual perceptual priming.
It proves definitively that perceptual priming lives entirely within the sensory cortices, operating completely independently of the explicit memory network.
Next on the implicit list is skills and habits.
This takes us away from the cortex and dives deep into the basal ganglia, specifically a structure called the striatum. This is the realm of reinforcement learning.
The striatum is absolutely essential for incrementally learning which actions are useful and with contexts based on continuous environmental feedback.
When you are learning the motor coordination to ride a bike or play a complex scale on the piano,
Or even learning complex probabilistic cognitive rules, like predicting the weather based on subtle cues where choice is only rewarded 70% of the time, you are relying on the striatum.
it is constantly adjusting the synaptic weights of your motor and cognitive circuits based on dopamine reward signals.
And to illustrate this deeply, we have another powerful clinical inverse to patient HM, which we see in patients suffering from Parkinson's disease.
Parkinson's disease is neurobiologically characterized by the profound degeneration of dopaminergic neurons in the substantia nigra.
Right.
Yeah.
Yes, neurons that project directly into the striatum. Because the striatum is literally starved of dopamine, the habit learning and probabilistic feedback loops begin to fail.
So early in the progression of the disease, a Parkinson's patient presents with the exact inverse cognitive profile of Henry Molaison.
Their explicit episodic memory for what happened earlier in the day is remarkably preserved, but they severely struggle to acquire new implicit motor habits or learn from trial and error probabilistic feedback.
Now, I want to point out a crucial nuance here, because in science, categories are helpful tools, but they aren't impenetrable brick walls.
Right.
We say implicit memory is separate from the hippocampus, but we have something called statistical learning that really blurs that clean dividing line.
It does, and it shows how dynamic the brain really is. Statistical learning is your brain's incredible ability to extract hidden regularities or complex grammars from a sequence of events entirely without your conscious awareness.
For instance, think about how an infant learns language.
An infant listening to adults speak automatically learns which syllables tend to follow other syllables, allowing them to intuitively find word boundaries in a continuous stream of sound.
They aren't consciously trying to map out linguistic probability.
Exactly. However, advanced studies show that when statistical learning requires linking highly complex relational sequences across time, the medial temporal lobe, the hippocampus actually engages to help extract those relational structures.
Even if the subject remains totally unaware of the pattern.
Yes. So implicit does not strictly equate to non-hippocampal in every single instance.
Right. The brain doesn't read our textbooks. It just uses whatever circuits get the job done in the moment. Let's move further down the evolutionary ladder to the most foundational, ancient forms of implicit memory.
Associative and non-associative learning.
Associative learning is exactly what it sounds like. The organism learns a predictive relationship between events. The two massive pillars here are classical conditioning and operant conditioning.
Classical conditioning is cue outcome learning. You learn that a previously neutral stimulus perfectly predicts a biologically significant event.
The classic example being Pavlov's dog learning that the bell predicts the food right.
Exactly. Or in fear conditioning, a harmless tone is paired with a mild electrical shock.
Very quickly, the animal learns that the tone perfectly predicts the shock.
And soon the tone alone will elicit a massive defensive freezing response. And the amygdala is the absolute central hub for this mechanism.
The amygdala integrates the auditory sensory information of the tone with the somatosensory pain signal of the shock.
It assigns biological significance to the tone and orchestrates the massive autonomic defensive response. Heart rate spikes, stress hormones flood the system.
While classical conditioning is about predicting what the world is going to do to you, Operant conditioning is about you changing the world.
It is action-consequence learning.
If an animal presses a lever and gets a food pellet, the probability of pressing the lever again increases. The behavior is reinforced by the consequence.
And finally, we have non-associative learning, which involves a change in response to just a single stimulus, not a relationship between two things.
Habituation is when your nervous system gradually decreases its response to a harmless, repetitive stimulus.
Think about the hum of an air conditioner in a room. After a few minutes, your brain actively tunes it out so you don't waste biological resources reacting to it.
Sensitization is the exact opposite.
If you experience a highly noxious, startling event, your nervous system becomes hyper-aroused, and you will suddenly overreact to even mild stimuli, like a gentle tap on the shoulder.
And it is so vital to understand that all of these learning mechanisms, from the simplest habituation of an air conditioner to the vast complexities of the hippocampus building an episodic memory, did not evolve in a sterile mathematical vacuum.
They evolved in brutal, unforgiving ecologies to solve very specific survival problems.
This brings us to Section 5, Biological Constraints and the Adaptive Errors of Memory.
The idea that the brain is a blank slate, a universal association machine that can learn any two things equally well as long as they occur together, is biologically false.
The brain is highly constrained and prepared by millions of years of evolution to learn specific relationships that matter for survival.
The ultimate proof of this biological preparedness is taste aversion.
If you think about classical conditioning rules, the tone and the shock have to happen almost simultaneously for the brain to link them.
But taste aversion totally breaks the standard rules of timing. It completely violates the standard temporal rules of associative learning.
An animal, or a human for that matter, can eat a novel food and then hours later become violently ill.
Despite the massive time delay between the stimulus and the consequence, the brain will instantly and permanently associate the specific taste of that food with the illness.
The animal will avoid that food for the rest of its life. Ecology demands this constraint.
In the wild, the toxic effects of a poisonous berry don't hit you immediately. They hit you after digestion. If the brain required immediate feedback to learn taste aversion, the species would go extinct.
The biology dictates the learning rules. Here's where it gets really interesting, and I want you to lean in for this part.
Yes, this is crucial.
When we finally understand that memory is an evolutionary survival tool, we have to completely reevaluate what we consider to be bugs in the system. The text outlines the 7 imperfections of memory.
And we need to reframe these not as failures of a broken machine, but as highly adaptive biological features. Let's really dig into these. Number 1 is transience.
Transience is the simple weakening or loss of a memory over time. The details fade, the edges blur, the trace becomes inaccessible.
We usually view this as a frustrating failure, but transience is vital. If your brain retained a perfectly vivid, high-resolution memory of every single place you have ever parked your car in your entire life, your mind would be paralyzed by massive, irrelevant clutter when trying to find your car today.
Forgetting outdated, useless information is an active, adaptive biological process that prevents cognitive overload.
We actually have a terrifying, real-world example of what happens when transience fails. The famous Russian neurologist Alexander Luria studied an mnemonist named Shereshevsky.
This man literally could not forget. He had an impossibly vast, highly detailed episodic memory, and it completely ruined him.
It was a deeply debilitating condition because Shereshevsky could remember every single micro detail of every specific object he ever saw. His brain was completely incapable of abstraction or generalization.
He struggled to understand basic concepts or metaphors because his mind was perpetually drowned in a tsunami of useless, hyperspecific details.
Forgetting is what allows us to abstract to find the pattern and to pull meaning from the noise.
Number 2 on the list of imperfections is absent-mindedness, and we all experience this constantly.
We do. Absent-mindedness is not a failure of memory storage. It is a failure of attention at the exact moment of encoding.
This is the classic losing your keys scenario. You walk into your house and put your keys down on the counter, but your working memory is actively occupied thinking about an anxious email you have to send.
Because your prefrontal attention wasn't placed on the physical action of dropping the keys, the information never even entered the encoding pipeline.
The memory of where the keys are doesn't exist to be forgotten. It was never written.
Number 3 is blocking. This is the one that drives people crazy.
Blocking is a temporary failure of retrieval. The information is fully encoded. It is successfully stored in the long-term tissue, but you cannot access it in that precise moment.
The tip of the tongue phenomena is the best example. The actor's name, you can picture their face,
Yeah.
what movies they have been in. But the specific phonological pathway to the name is temporarily blocked by competing neural signals.
It proves that storage and retrieval are entirely distinct mechanisms. The file is there, but the search function is temporarily jammed.
Number 4 is misattribution. This is a big one, especially in the era of information overload.
Misattribution is assigning a perfectly accurate memory to the completely wrong source.
You might clearly remember a highly specific, complex fact about the economy, but you falsely attribute it to a peer-reviewed article you read in a reputable journal, when in reality you heard it from a random person on the bus.
Your brain successfully stored the semantic content, the fact, but it decoupled the content from the episodic context in which it was learned.
And that vulnerability in the system leads directly to number 5, suggestibility, which is absolutely terrifying when you think about the legal system and eyewitness testimony.
Suggestibility is the phenomenon where external misinformation is actively incorporated into your own personal memory of an event.
Because retrieval is reconstructive. The way a memory is probed can literally rewrite the trace. The pioneering psychologist Elizabeth Loftus demonstrated this beautifully and terrifyingly.
If a police officer asks a witness a leading question, say how fast was the car going when it smashed into the truck versus when it bumped into the truck, the mere use of the word smashed can alter the witness's reconstructive process.
Weeks later, they will vividly reconstruct the memory to include shattered glass on the road that was never actually there. The external suggestion physically edits the internal biological file.
Number 6 is bias.
Bias shows us that we always unavoidably view the past through the lens of the present. Your current beliefs, your current emotional state, and your current identity actively reshape how you interpret and reconstruct past events.
If you are currently deeply angry at a friend, your brain will preferentially retrieve and reconstruct memories of past interactions that highlight their flaws.
Subtly editing the tone of those memories to justify your current emotional state. We rewrite our history to maintain a consistent narrative of who we are today.
And finally, number 7 is persistence.
Persistence is the inability to forget. It is the occurrence of unwanted, highly intrusive memories that repeatedly and aggressively force their way into conscious awareness.
In its extreme clinical form, this is a hallmark of PTSD following severe trauma.
It is incredibly disabling for the individual, but from a cold evolutionary perspective, persistence has a brutal logic.
If an organism survives a near-death encounter with a predator, the brain ensures that the organism never, ever forgets the environmental cues associated with that near-death experience.
Prioritizing future survival over psychological comfort.
Understanding that memory is an active, reconstructive, highly adaptive system completely changes how we should approach absorbing new information. Which brings us to our outro and actual practical educational implications of this entire deep dive for you, the listener.
If you want to truly learn something, if you want to force your brain to undergo the heavy metabolic cost of long-term biological change, passive exposure will fail you every single time.
You cannot just highlight a test book or reread your notes.
Durable learning requires deep encoding. You have to actively manipulate the meaning of the information.
It requires retrieval practice, right?
Yeah, the very act of forcing yourself to recall information without looking at the source actually strengthens the neural pathways much more than just rereading it.
And it requires spaced repetition, allowing the cellular and systems consolidation processes time to operate and physically restructure the network between study sessions.
True learning is about conceptual binding. Just as the hippocampus performs relational binding, stitching the visual and auditory pieces into an episode, you have to consciously bind new concepts to the things you already know to create a robust semantic network.
Let's bring this entire chapter together. Let's take all these disparate complex systems and put them into one single, unified, everyday scenario to show how they orchestrate your reality.
Imagine you are walking into a busy cafe to meet an old friend you haven't seen in years.
Okay, let's forensically walk through the circuits.
As you open the heavy wooden door, your visual association cortices are instantly processing the shape of the tables, the lighting, and scanning for the face of your friend.
Your auditory cortices process the clatter of plates, the background jazz music, and eventually the pitch of their voice calling your name.
Your semantic memory instantly activates your conceptual knowledge of what a cafe is, the social rules of how to behave in one, and the factual history of who your friend is and what they do for a living.
Simultaneously, your episodic memory uses the hippocampus to instantly retrieve the memory of the last time you saw them, providing the vital social context for the current meeting.
Your working memory is actively maintaining the thread of the conversation, holding on to their sentences on the cutting board so you can formulate an intelligent, relevant reply.
Your amygdala and emotional systems are evaluating their microexpressions, assigning effective value to the interaction. Are they happy to see you? Are they stressed?
And underneath all of that conscious high-level awareness, your procedural memory and your basal ganglia are automatically coordinating the insanely complex motor sequence of navigating through a crowded room, pulling out a chair, sitting down, and lifting a hot, full cup of coffee to your mouth without spilling a single drop.
A single unified ordinary moment of human experience is a breathtaking illusion of simplicity.
It is generated by the seamless, synchronized firing of multiple, highly specialized memory systems distributed across the entire brain.
And later, when you try to remember that coffee date next week, your brain will reconstruct it. It will drop some details, it will infer others, and it will be deeply colored by how you feel about the friend tomorrow. But the memory will serve its purpose.
It is truly breathtaking when you realize what's happening under the hood.
And this deep dive serves as our conceptual map for the rest of this unit. We have established the systems-level framework. Next up in Chapter 53, we are going to zoom way in and explore the cellular and molecular storage mechanisms of implicit memory.
And then in Chapter 54, we will tackle the intricate biology of explicit memory inside the hippocampus itself. We've established the macroarchitecture. Next, we look at the microscopic bricks and the mortar.
As we wrap up, I want to leave you with one final provocative thought to chew on.
If we connect all of this to the bigger picture, if our memory systems evolve strictly to help us survive by simulating and predicting the future, and if every single act of retrieval actively alters the memory trace, we have to ask a very difficult philosophical question.
What is it?
How much what you currently consider your authentic past, the foundational narrative of who you think you are, is actually just a highly edited, personalized survival manual written by your brain to keep you safe tomorrow?
The brain does not merely record your life like a librarian quietly filing away the days. It is a loom constantly weaving and reweaving the threads of your experience based on the needs of the present.
You are not a static archive. You are a living, breathing transformation.
Thank you for joining us on this incredible journey through the biology of memory. Keep questioning, keep learning, and we will see you for the next deep dive.